DNA replication. First step, initiation. DNA protein initiates replication in E. coli at ORIC. DNA protein binds to the 9-mer sequence in ORIC, forming an initial complex. Binding of DNA to the ORIC 9-mers facilitate the initial strand separation or melting of E. coli double helix, which occurs at the ORIC 13-mers. This process requires ATP and forms open complex. Further melting of the two strands of the E. coli chromosome to generate unpaired template strands is mediated by DNA B protein, a helicase. One example of DNA B, a hexamer of identical subunits, clamps around each of the two single strands in the open complex formed between DNA A and ORIC. This binding requires ATP and DNA C protein. Helicase moves along a DNA duplex utilizing the energy of ATP hydrolysis to separate the strands. In E. coli, the separated strands are inhibited from subsequently re-annealing by a single-strand binding protein, SSB protein, which binds to both separated strands. DNA polymerase can only elongate existing primase strands of DNA or RNA. The primers used during DNA replication in both prokaryotes and eukaryotes are short RNA molecules whose synthesis is catalyzed by the RNA polymerase primase. After the bound primase synthesized short primers RNA is complementary to both strands of duplex DNA. They dissociate from the single-stranded template. In elongation, DNA polymerase catalyzes the step-by-step addition of deoxyribonucleotide units to DNA chain. The chain elongation reaction catalyzed by DNA polymerase is a nucleophilic attack by the 3' hydroxyl group of the primer on the innermost phosphorus atom of the deoxyribonucleoside triphosphate. A phosphodiester bridge forms with a concomitant release of pyrophosphate. The subsequent hydrolysis of pyrophosphate by pyrophosphatase, a ubiquitous enzyme, helps to drive the polymerization forward. Elongation of the DNA chain proceeds in the 5' to 3' direction. Here we could see the picture on figure. The structure of the DNA chain and mechanism of growth by addition of deoxyribonucleoside triphosphate at the 3' end of the chain. At each growing fork, one strand called the leading strand is synthesized continuously from a single primer on the leading strand template and grows in the 5' to 3' direction. Growth of the leading strand proceeds in the same direction as movement of the growing fork. Synthesis of the lagging strand is more complicated because DNA polymerase can add nucleotides only to the 3' end of the primer to growing DNA strands. Movement of the growing fork unveils the template strand for lagging strand synthesis in the 5' 3' direction. After 1000 to 2000 nucleotides of the leading strand have been replicated, the first round of discontinuous strand synthesis on the lagging strand can begin. Short pieces of DNA called Okazaki fragments are repeatedly synthesized on the lagging strand template. Size of the Okazaki fragments are 1000 to 2000 nucleotides in bacterial cells and 100 to 200 nucleotides in eukaryotic cells. Mode of replication in which one new strand is synthesized continuously while the other is synthesized discontinuously is known as semi-discontinuous replication. Parental strand, leading strand, pole 3 complex, DNA B helicase SSB, primase, lagging strand. Synthesis of leading and lagging strands of DNA. The leading strand is synthesized continuously in the direction of replication focal movement. The lagging strand is synthesized in small piece or Okazaki fragments backward from the overall direction of replication. The Okazaki fragments are then joined by the action of DNA helicase. The primase, which remain associated with the DNA B helicase in the primosome, makes an RNA primer, which is then extended by DNA polymerase 3. This is the same DNA polymerase 3 complex that is synthesized the leading strand complex, the complex comprising in effect 2 copies of the polymerase. The primase, which remain associated with the DNA B helicase in primosome, makes an RNA primer, which is then extended by DNA polymerase 3. This is the same DNA polymerase 3 complex that is synthesizing the leading strand copy, the complex comprising in effect 2 copies of the polymerase. It is not 2 complete copies because there is only a single gamma complex. The main function of the gamma complex is to interact with beta subunit, the sliding clamp, and hence control the attachment and removal of the enzyme from the template. The main function of the gamma complex is to interact with the beta subunit, the sliding clamp, and hence control the attachment and removal of the enzyme from the template. A function that is required primarily during lagging strand replication and the enzyme has to attach and detach repeatedly at the start and end of each Okazaki fragment. Although the two core polymerase molecules are linked by the tau subunit dimer, they are oriented in opposite directions. So this picture depicts E. coli replication proteins at a growing fork. DNA B helicase moves along the lagging strand template toward its 3' end. Once again, DNA B helicase moves along the lagging strand template toward its 3' end, thereby melting the duplex DNA at the fork. The leading strand polymerase core 1 together with its subunit clamp synthesizes the leading strand continuously. The second core 2 synthesizes the lagging strand discontinuously as a Okazaki fragments, and the two core pole molecules are linked via a dimeric protein. So direction of growth of lagging strand, primer, core 2, pole 3, leading strand, direction of growth of leading strand, beta clamp, core 1, pole 3, and helicase. As each Okazaki fragment formation completes, the RNA primer of the previous fragment is removed by the 5' 3' exonuclease activity of DNA polymerase 1. This enzyme also fills in the gaps between the lagging strand fragment, which then be ligated together by DNA ligase. The joining of Okazaki fragments requires an enzyme DNA ligase that catalyzes the joining of the ends of two DNA chains. This enzyme catalyzes the formation of a phosphodiester bond between the 3' OH group at the end of the DNA chain and the 5' phosphate group at the end of the other. An energy source is required to drive this thermodynamically uphill reaction. In eukaryotes and archaea, ATP is the energy source. In bacteria, NAD+ typically plays this role. ATP, NAD+ function as the AMP donor, and PAP NMN is released. E plus ATP plus NAD+ gives EAMP plus PPI or NMN. EAMP P plus 5' DNA, E plus AMP P 5' DNA 3' plus AMP P 5' DNA O 3' O P 5' DNA plus AMP gives DNA 3' OH plus P 5' DNA ATP or NADP plus DNA 3' O P 5' DNA plus AMP plus PPI or MMN. As in E. coli, completion of lagging strand synthesis require removal of DNA primer from each Okazaki fragment. There appears to be no eukaryotic DNA polymerase with the 5' to 3' exonuclease activity. So process is different from bacterial cells. In eukaryotes, most of the RNA component of the primer is removed by RNase H, which can degrade the RNA part of the base paired RNA-DNA hybrid, but cannot cleave the phosphodiester bond between the last ribonucleotide and the first deoxyribonucleotide. The last ribonucleotide is removed by the flap endonuclease. So remember RNase H and flap endonuclease. So RNase H removes the primer up to the last ribonucleotide. RFEN1 removes that last ribonucleotide. RNA ligase links the two DNA fragments. Primer removal in eukaryotes, RNase H model. Last one is removed by flap endonuclease, FEN1, flap endonuclease. Next is termination. Bacterial genomes are replicated bidirectionally from a single point, which means that the two replication forks should meet at a position diametrically opposite the origin of replication on the genome map. Restriction of genome terminus region containing single copies of about 23 base pair sequence is called for terminus sequence. 7 of these have been identified in the E. coli genome, with each one acting as a recognition site for a sequence-specific DNA binding protein called TUS, terminus utilization substance. When bound to terminator sequence, a TUS protein allows a replication fork to pass if the fork is moving in one direction, but block process if the fork is moving in the opposite direction around the genome. The directionality is set by the orientation of the test protein on the double helix. When approached from one direction, thus blocks the passage of DNA B helicase, which is responsible for progression of the replication fork. But when approaching from the other direction, DNA B is able to cross the test protein. ORIC, TRE, TRD, TRA, TRC, BFG portions. ORC, termination of chromosome replication in E. coli. The replication forks that start at ORIC can transverse TRA and TRB in only one direction opposite to that indicated by the arrows. The orientation of the termination sequence and hence of the bound test protein in the E. coli genome is such that both the replication fork become trapped within a relatively short region on the opposite side of the genome to the origin. This ensures that termination always occur at or near the same position. Then proofreading. DNA replication is amazingly accurate with only about one error for every billion bases incorporated. This accuracy is necessary to keep the mutation load at a tolerable level, especially in large genomes such as those of mammals, which contain about 3 into 10 raised to 9 nucleotide bases. DNA proofreading involves scanning the termini of messenger DNA chains for error and correcting them before continuing chain extension. This process is carried out by a 3' 5' exonuclease activity that is built into DNA polymerase. When a template primer DNA has a terminal mismatch, an unpaired or incorrectly paired base or sequence of bases at the 3' end of the primer, the 3' 5' exonuclease activity of the DNA polymerase clip off the unpaired base or bases when an appropriately base paired terminus is produced. The 5' 3' polymerase activity of the enzyme begins resynthesis by adding nucleotides to 3' end of the primer strand. The synthesis of Okazaki fragments is a key element in DNA replication. Each Okazaki fragment is initiated by an RNA primer. How does these RNA primers are produced? How are they removed from Okazaki fragments? How are the gaps that result from their removal filled? How are the segments covalently joined to form continuous lagging strands? The RNA primer is made complementary to the DNA template strand in the direction by an enzyme called primase using ATP, UTP, CTP, and GTP as precursors. After an Okazaki fragment has been synthesized by the DNA polymerase III complex, the RNA primer is removed by the 5' 3' exonuclease activity of DNA polymerase I. The gap that results is filled by DNA polymerase I activity using the 3' end of the Okazaki fragment as a starting point. The elongated Okazaki fragments are covalently joined to two continuous lagging strands by the action of DNA ligase. Some E. coli mutants contain defective DNA ligase. When these mutants are exposed to 13H3H deuterium, proterium, tritium labeled thymine, and the DNA product is sedimented on an alkaline sucrose density gradient. Two radioactive bonds appear, one corresponding to the high molecular weight fraction and one corresponding to low molecular weight fraction. During replication of the DNA duplex, the leading strand is replicated continuously and the lagging strand is replicated in short fragments called Okazaki fragments, which are then spliced together by means of DNA ligase. Mutants with defective DNA ligase produce DNA duplex in which one of the strands remain fragmented. Consequently, when this duplex is denatured by the alkaline conditions of the sucrose gradient, sedimentation results in one fraction containing the intact single strand, the high molecular high band, and the fraction containing an unspliced fragments, low molecular weight band. And telomerase replication replicates the ends of chromosome. All eukaryotic chromosomes end in telomeres. The replication process, which generates multiple copies of the telomeric repeats, is independent of the normal DNA replication machinery, which fails to replicate the extreme 5' end of the chromosome strand. A special enzyme called telomerase is employed to catalyze the replication at the telomere. So, the replication process, which generates multiple copies of the telomeric repeats, is independent of the normal replication machinery, which fails to replicate the extreme 5' end of the chromosome strand. A special enzyme called telomerase is employed to catalyze the replication at the telomere. Telomerase enzyme is a ribonucleoprotein, contains RNA and protein. It resembles reverse transcriptase enzymes that synthesize DNA using an RNA template. Telomerase recognizes the tip of the G-rich strand of an existing telomere DNA repeat sequence and elongates it in 5' to 3' directions. The telomerase synthesizes a new copy of the repeat. Telomerase recognizes the tip of G-rich strand of an existing telomere DNA repeat sequence and elongates it in the 5' to 3' direction. The telomerase synthesizes a new copy of the repeat using an RNA template that is a component of the enzyme itself. After several rounds of extension of the parental DNA strand by telomerase, replication of the lagging strand at the chromosome end can be completed by using this extension as a template for synthesis of the complementary strand by a DNA polymerase molecule. The mechanism just described ensures that the 3' end at each telomere is always slightly longer than the 5' end with which it is paired, leaving a protruding single-stranded end. So parental strand, incomplete newly synthesized lagging strand, telomerase binds. And direction of telomerase synthesis, telomerase extends 3' end, telomerase with bound RNA template, then completion of lagging strand by DNA polymerase, then DNA polymerase. So mechanism of action of telomerase. The single-stranded 3' terminus of a telomere is extended by telomerase, converting the inability of the DNA replication mechanism to synthesize the extreme terminus of linear DNA. Telomerase elongates this single-stranded end by a reverse transcription mechanism. The telomerase contain an RNA template that base pairs to the 3' end of the lagging stranded template. The telomerase catalytic site then adds deoxyribonucleotides using the DNA molecule as a template. Next is about rolling circle replication. Replication via theta forms is not the only method by which circular DNA molecules can replicate their genetic information. Another method is rolling circle replication that generates linear copies of genomes rather than circular copies. The rolling circle model of replication. A replication fork proceeds around a circular template for an indefinite number of revolutions. Then the DNA strand, newly synthesized in each revolution, displaces the strand synthesized in the previous revolution, giving a tail containing a linear series of sequences complementary to the circular template strand. Consider a circular molecule of double-stranded DNA with a nick in one of the two phosphodiester backbone. A nick opens one strand and then the free 3' OH end generated. As long as there is free 3' OH end, this can serve as a primer for DNA polymerase. When the 3' OH is extended, the 5' end can be displaced in a manner analogous to the strand displacement reaction. Extension of 3' OH end is also analogous to leading strand synthesis. The displaced strand can in turn serve as a template for replication as long as a suitable primer is available. Synthesis on this strand is analogous to lagging strand synthesis. The template is circular duplex DNA. Initiation occurs on one strand. Elongation of growing strand displaces old strand. After one revolution, displaced strand reaches unit length. The rolling circle method of replication. If synthesis continues in this manner, the consequence of this mechanism of replication can be production of concatemer copies of circular molecule. As a result, multiple copies of a genome are produced. A rolling circle model of replication is seen both during replication of bacteriophage lambda, where rapid production of many copies of genome is desired, and in the replication of bacteriophage M13, where only a single copy is produced each time. So replication of mitochondrial DNA. Some smaller circular molecules such as mitochondrial and chloroplast genome use a slightly different process of replication. Replication starts at a specific origin in the circular duplex DNA, but mitochondrial DNA use different origin of sequence to initiate replication of each DNA strand. Initially, only one of the two parent strands is used as a template for synthesis of a new strand. Synthesis proceeds for only a short distance, displacing the original complementary strand which remains single-stranded. This pattern of replication generates displacement or D-loop. A single D-loop is found as an opening of 500 to 600 bases in mammalian mitochondria. Some mitochondrial DNAs possess several D-loops, which reflects the presence of multiple origins. The similar mechanism is employed in chloroplast DNA. Replication of the complementary strand is initiated when its origin is exposed by the movement of the first replication fork. So H strand is there, L strand is there. DNA synthesis initiated by origin on H strand. Then synthesis of new L strand creates a D loop by displacing parental strand. Then D loop expands. Later, when displaced strand passes origin on L strand, synthesis of new H strand starts. Replication of mammalian mitochondrial DNA. Replication starts at a specific origin in the circular duplex DNA. Initially, only one of the two parental strands, the H-stranded mammalian mitochondrial DNA, is used as a template for synthesis of new strand. Synthesis proceeds for only a short distance, displacing the original partner L strand, which remains in single-stranded. There is separate origins for L and H strand. Then recombination. Recombination, large-scale rearrangement of DNA molecule, was first recognized as the process responsible for crossing over and exchange of DNA segments between homologous chromosomes during meiosis of eukaryotic cells, and was subsequently implicated in the integration of transferred DNA into bacterial genomes after conjugation, transfection, or transformation. The genetic recombination events fall into two general classes. Homologous recombination. Recombination which involves genetic exchanges between any two homologous DNA molecules or segments of the same molecule that share an extended homology. Then site-specific recombination. Recombination binds the two dsDNA molecules that have only two regions of nucleotide sequence similarity. A different type of event called transposition, which is related to the process of recombination, allows one DNA sequence to be inserted into another without relying on sequence homology. It provides a means by which certain elements move from one chromosomal location to another. Then homologous recombination, general recombination. Homologous recombination is the most important version of recombination in nature, being responsible for meiotic crossover and the integration of transferred DNA into bacterial genome. It involves the reciprocal exchange of sequence of DNA, the Holiday model for homologous recombination. An appealing scheme for homologous recombination was proposed by Robin Holiday in 1964. The Holiday model describes recombination between two homologous double-stranded molecules, one with identical or nearly identical sequence, but is equally applicable to two different molecules that share a limited region of homology, or a single molecule that recombine with itself because it contains two separate regions that are homologous with one another. Endonuclease nicking, then strand displacement, then ligation, and Holiday junction. Then ligation, Holiday junction, and branch migration will be taking place. The Holiday model for homologous recombination. Single-strand nicks are introduced at the same position on both parental molecules. The nicked strands then exchange by complementary base pairing, and ligation produces a cross-strand intermediate called a Holiday junction. So the general feature of the model is formation of a heteroduplex resulting from the exchange of polynucleated segments between the two homologous molecules. The heteroduplex is initially synthesized by base pairing between each transferred strand and the intact polynucleotide of the recipient molecule, this base pairing being possible because of the sequence similarity between the two molecules. Subsequently, the gaps are sealed by DNA ligase, giving a Holiday structure. This structure is dynamic, branch migration resulting in exchange of longer segments of DNA if the two helices rotate in the same direction. Separation or resolution of the Holiday structure back into individual double-stranded molecules occur by cleavage across the branch point. This is the key to the entire process because the cut can be made in either of two orientations, as become apparent when the three-dimensional configuration or chi form of the Holiday structure is examined. These two cuts are very different results if the cut is made left-right across the chi form. This is the key to the entire process because the cut can be made in either of the two orientations, as become apparent when the three-dimensional configuration or chi form of the Holiday structure is examined. These two cuts are very different results if the cut is made left-right across the chi form. Then all that happens is that a short segment of polynucleotide corresponding to the distance migrated by the branch of the Holiday structure is transferred between the two molecules. On the other hand, an up-down cut results in reciprocal strand exchange, double-stranded DNA being transferred between the two molecules so that the end of the one molecule is exchanged for the end of the other molecule. This is the DNA transfer chain and crossing over. So duplex separation, 180 degrees Celsius rotation, holiday structure or chi form, Nick recombinant duplexes. So resolution of holiday structure. Then Michelson-Rading modification. Michelson-Rading modification explains the way in which Michelson-Rading modification explains the way in which two double-stranded molecules interact at the beginning of the process to produce the heteroduplex. In Holiday model, the two molecules line up with one another and single-stranded nicks appeared at equivalent portions in each helix. This produced free single-stranded ends that could be exchanged, resulting in the heteroduplex, but not explains how the nicks occurred at precisely the same position on each molecule. The Michelson-Rading modification proposes a more satisfactory scheme whereby a single-stranded nick occurs in just one of the double helixes, the free end that is produced invading the unbroken double helix at the heteroduplex position and displaying one of the strands forming a D loop. Subsequent cleavage of the displaced strand at the junction between its single strand and base pairing regions produce the heteroduplex. So nicking of one parental DNA displacement of nicked strand, displaced DNA strand invades another parental molecule, cleavage of loop pairing with first parental molecule. So initiation of recombination by nicking only on parental molecule. The nicked DNA strand inverts the other parental molecule by homologous base pairing, thereby displacing a single-stranded loop of DNA. This loop is then cleaved and pairs with the first parental molecule, yielding a cross-stranded holiday junction. Proteins involved in homologous recombination in E. coli. Homologous recombination occurs in all organisms, but the nature of the events involved in homologous recombination between DNA molecules is first described in bacterial systems. Three different recombination systems, REC-BCD, REC-CE and REC-CF have been described, with REC-BCD apparently the most important in the bacteria. Recombination is initiated by the REC-BCD enzyme, which has both nuclease and helicase activities. Its role in recombination is to provide a single-stranded region with a free 3' end. The REC-BCD enzyme binds to linear DNA at a free broken end and moves inward along the double helix, unwinding the double helix and degrading the released single strand with 3' end in a reaction coupled to ATP hydrolysis. The activity of the enzyme is altered when it interacts with sequence referred to as chi. The chi sequence occurs naturally in E. coli DNA about once every 5 to 10 kilobase pairs. When REC-BCD enzyme reaches the chi site, it recognizes chi site in single-stranded form. This causes the enzyme to pause. Then it cleaves the top strand of the DNA at a position between 4 and 6 bases to the right of chi. This process creates a ssDNA with 3' end. Recognition of the chi site by REC-BCD enzyme causes the REC-D subunit to dissociate, as a result of which enzyme loses its nuclease activity. However, it continues to function as a helicase. Then chi site REC-BCD binds to an end of dsDNA. Then REC-BCD unwinds dsDNA and degrades ssDNA with the 3' end. After recognition of the chi site, it loses REC-D subunit and retains only the helicase activity. So REC-BCD binds to DNA at a double-stranded end. As REC-BCD advances, it degrades released DNA single strand with a 3' end. When it reaches the chi site, it then cleaves the top strand of the DNA at a position between 4 and 6 bases to the right of chi. Recognition of the chi site causes the REC-D subunit to dissociate, as a result of which enzyme loses its nuclease activity. The overall result of this interaction is to generate single-stranded DNA with 3' end at the chi sequence. This is the substrate for recombination. The free single-stranded end, according to the Michelson-Rading modification, invades the intact partner, the circular E. coli genome, which is mediated by the REC-A protein, which forms a protein called a coated DNA fragment. Then that is able to invade intact double helix and set up D loop. Branch migration is catalyzed by the RUVA and RUE proteins, both of which attach to the branch point of the holiday structure. the G protein also has a role in branch migration, but it is not clear if it is in conjugation with RUVAB or as a part of the alternative mechanism. The RUVAB complex can cause the branch to migrate as fast as 10 to 20 base pair per second. Branch migration from exchangeable site determines the length of the region of hybrid DNA. When branch migration has ended, the RUVAB complex detaches and is replaced by two RUVC proteins which carry out the cleavage that resolves the holiday structure. A common tetranucleotide sequence ATTG provide a hotspot for RUVC to resolve the holiday junction. Then next is about double strand break model. This model is similar in many ways to the model discussed previously, but initiation is by a double strand break in one of the two DNAs. Initiation occurs by a double strand peak break in one DNA molecule. The recipient DNA is resected from the break by an exonuclease that leaves 3' OH overhangs. One of these overhangs can invade the homologous region and the other donor duplex. This is called single strand inversion. The formation of heteroduplex DNA generates a D loop in which two strands of the donor duplex is displaced. Because the inverting strands and with the 3' termini, they can serve as a primer for new DNA synthesis. Elongation from these DNA ends using the complementary strand in the homologous duplex as a template serves to regenerate regions of DNA that were destroyed during the process of the strands at the break site. The two holiday junctions found in the recombination intermediates generated by this model move by branch migration and ultimately are resolved to finish recombination. So double stranded break, recipient donor, strand inversion of 3' and strand extension. Then branch migration and formation of two holiday junctions, double stranded break model of homologous recombination. Then site-specific recombination. Next one is site-specific recombination. A region of extensive homology is not a prerequisite for recombination. The process can also be initiated between the two DNA molecules that have only very short sequence in common. This is called site-specific recombination. Integration of bacteriophage lambda DNA into E. coli genome involves site-specific recombination. Integration occurs by site-specific recombination between the ATT sites, one at the lambda genome and on the E. coli chromosome, which have at their center an identical 15 BP sequence. The bacterial attachment site called ATTB, consisting of the sequence components BOB-, the attachment site on the phage ATTP consists of the components POP-, the sequence O is common to ATTB and ATTP. This is called core sequence and the recombination events occur within it. The recombination event is catalyzed by a specialized type 1 teloisomerase called integrase. The enzyme makes a staggered double-stranded cut at equivalent positions in the delta, lambda and bacterial ATT sites. The two short single-stranded outhangs are then exchanged between the DNA molecule, producing a holiday junction which migrates a few base pairs along the heteroduplex before being cleaved. Thus, cleavage, providing that it is made in the appropriate orientation, resolves a holiday structure in such a way that the lambda DNA becomes inserted into the E. coli genome. Lambda DNA, ATT site, bacterial DNA, integration of the bacteriophage lambda genome into E. coli chromosomal DNA. Both the lambda and E. coli DNA have a copy of ATT site, each one comprising an identical central sequence called O and flanking sequence P and P' for the phage ATT site, or P and P' bacterial ATT site. Recombination between the O regions integrate with the lambda genome into the bacterial DNA.